Hello boys, welcome back to another session of video tutorials. This is meant for class 11th biology students and this is meant for 17th July 2020. From our memory we understand that we have been discussing about a chapter called Cell, the Unit of Life, and we were talking about various cell organelles and their functions, and we were discussing in our previous session a very important cell organelle that helps in protein synthesis, that is ribosomes. And in this session we are going to discuss about another important organelles which are small in nature but very important as far as their function is concerned. Now in this session we are going to talk about centrosome and centriole. What is centrosome? See, within the cell you can spot here the centrosome. You got two dots and there are astral rays just like sun rays around it. So it's generally present in animal cell, but in some algae, fungi, moss, fern, the cells have centrioles near the nucleus. This is the nucleus. Near the nucleus centrioles are there. It has a pair of dots, dot 1, dot 2, dot 1, dot 2 granules surrounded by transparent cytoplasmic centrospheres. Whole structure is known as centrosome. So this whole structure is centrosome, and centrioles have astral rays and are functional in cell division in formation of spindle. So during the cell division the chromosomes will be in pairs, and some chromosomes will be pulled up and some will be pulled down. We say north pole, south pole, and this pulling of chromosomes will be done by centrioles. And as you know, centrioles are very minute, submicroscopic, microtubular, subcylinder, found in almost all eukaryotic animal cells. Some fungi and plants with flagellated structures are also present in this. Now, what is the structure of a centriole? If we expand this and observe under electron microscope, we will see in the transfer section of centriole, a centriole possesses a wall of 9 peripheral fibrils. So you got 9 peripheral fibrils, remember. And fibrils are absent in center, forming 9 plus 0 arrangement. Remember this in exams. So centriole has got 9 plus 0 arrangement of peripheral fibrils, and each fibril is made up of 3 sub-fibers called triplet fibril. So 9 fibrils are there and each one has got 3. So 9 into 3. So this many 27 triplet fibrils will be there. From outside to inside, these 3 subfibers are named as C, B and A. The adjacent triplet fibrils are connected by C to A proteinaceous linkers. So the center of centriole possesses rod-shaped proteinaceous mass known as hub. So there is a central hub, and surrounding this you have got 9 proteinaceous strands developed towards the peripheral triplet fibrils, and these are called spokes. So you must remember that there will be spokes in centriole. And what are the functions? Centriole can be transformed into basal bodies which give rise to cilia and flagella. So centriole is giving birth to cilia and flagella, and centrioles help in during mitosis as well as meiosis. Cell division time, this will help, and that is the role of this. If centrioles are not to be present, then what happens? There won't be formation of haploid organism. Everything will be diploid only. So we would have lost our identity. That is the reason why it is so important. And you must remember 9 plus 0 organization of microtubules in centriole. Okay, and now we come to the next one, cilia and flagella. We said just now centrioles will give birth to cilia and flagella. And as you know, paramecium, recollect, you must have already seen paramecium under microscope, and that has got hair-like structures around the body. Those hair-like structures are called cilia. And euglena, you must have seen a whip-like structure, that's flagella. So basically these are cilia and flagella are organelles for mobility, movement. So unicellular organisms like paramecium, you are seeing here, the chlamydomonas, you can see the flagella, and these are responsible for movements. And cilia and flagella, they too have got a peculiar arrangement. When we cut the section TS of flagellum, we'll see the cilia and flagella are microscopic, contractile and filamentous processes of the cytoplasm, and they are capable of producing a current in fluid medium for locomotion. For movement, they are responsible. And they synthesize polypeptide by enclosing it in groove and the passage of substances. So both arise, that is cilia as well as flagella will arise from basal bodies and are similar in chemical composition. They have basically identical ultrastructure and they serve as for locomotion purpose. However, the two are distinguished by their number, size and mode of beating. Now here also, just like you have seen in centriole, in flagellum also you have got spokes. So you have got there it is 9 plus 0, here it will be 9 plus 2 arrangement. There is a central sheath, there is a spokes, there is a head and inner side arm. All this will be making the flagellum very strong. This. And now we move on to the next one. You can see the arrangement here. Cilia beat like tiny oars or pedals as in boat, and flagella bring about whip-like lashing movement. Both are made up of contractile protein, tubulin, remember this, tubulin, and form microtubules. The arrangement, as I said just now, in case of centriole it is 9 plus 0. Here it is 9 plus 2. You can see here 1, 2, 3, 4, 5, 6, 7, 8, 9 and 1, 2, 9 plus 2 arrangement. Two central microtubules surrounding by 9 peripheral ones. So the cilia and flagella appear to rise from the basal body. And cilia are short, of course they are very hair-like structures but numerous, and flagella is long and only 1 or 2. And the microbodies, these are very, very small microbodies, as the name suggests, they are very small. The spherical bodies and the single membrane bound organelles. And some of these bodies are associated with chloroplast, mitochondria, endoplasmic reticulum, Golgi body. So on the basis of enzymes present and a tissue in which microbodies are present, they are classified into peroxisomes. Now, what are peroxisomes? They contain enzymes for peroxide biosynthesis. Peroxide biosynthesis found in both plant and animal cells, generally in close association with endoplasmic reticulum, mitochondria and chloroplast. And these are discovered by Tolbert. So mainly they are for they have enzymes for peroxide biosynthesis. These are peroxisomes. And then you have got glycosomes. These glycosomes contain enzymes for beta oxidation of fatty acids and glycolate pathway. Acetyl-CoA produced by beta oxidation of fatty acids is metabolized in glycolate cycle to produce carbohydrates. Following this, they convert back into peroxisomes. So glycosomes, of course, they are discovered by beavers, and they are mainly for conversion of fats into carbohydrates. That much you remember. And sperosomes, and the sperosomes, of course, these are special spherical bodies, half unit membrane, contain fatty materials, act as center for synthesis and storage of fats. This is the most important one. So we will be studying in our next session further